Works well now.
Yes. Okay. My name is Teija Tenhola-Roininen, and I'm working in Luke in a genomics and breeding group. And I will talk about two projects where I'm using, or we have used, rye landraces. And why we have started to study these rye landraces, it's because rye can survive from different stresses better than other cereals because it has a very good this rooting system, and because it's a cross-pollinating grain, it has very high genetic diversity. And in Finland, all Finns eat rye bread, sourdough rye bread, and it has been used for centuries in Finland, and that's why our rye cultivation has a very long history. And during nowadays, many Finns wants to make bread themselves, and they want to do it from local varieties. And we know also that there are a lot of beneficial compounds in rye, and it's because of the many things, and also because this sourdough has these lactic acid bacteria and yeast. And also these days it's very important to have self-sufficiency in cereal production. And here are all those landraces which we have used in our studies, and these material came from the farmers' fields. So we wanted to use those landraces which are already in cultivation, and we got the seeds from Minna-Maria Kytöviita from University of Jyväskylä and also from Annika. And also we got some old rye varieties from boreal plant breeding. This Hankkias Jussi is one of them, and then Riihi. And we used also in the studies this Tvengsberg, which was from Annika, but it's originally from the Norway. And also we had Elvi and Sangaste, which are originally from Estonia. And we used as controls this modern population variety Reetta, which is belongs to Boreal. And then we had this LO7 inbred line, and rye hybrid Livado from KWS from Germany. And here is one of the projects where we have used these rye landraces. It's called RYSAS, and the coordinator of this project is Joachim Fromme from Germany, and also Bernd Hackauf is working with him. And Luke and Boreal plant breeding are also partners, and then there are in total 11 different partners in this project. And our goals in this RYSAS project has been to develop these semi-dwarf rye hybrids and then test them on the field, and then do this kind of simulation model for rye. And this part where we want to find out the genetic architecture of winter hardiness, there we have used these Scandinavian rye landraces, and also which includes also these Finnish ones. And then we wanted to also test this ergot contamination in this hybrid material, and also our goal is to increase selection speed in breeding process. And this is the whole project structure, and Finland is in 4 of those work packages involved, and in this work package 6 in molecular breeding part, we have used these Rye Landraces. And this work package 6 is called Genome-Based Breeding for Frost Tolerance. And we have genotyped this kind of rye diversity set. It included at least 50 different rye landraces, some of them Finnish ones. And in these analyses, there were 6 single plants used per population. And these analyses were done in Trade Genetics, Germany, and we used this kind of custom wheat-rye triticale SNP array for these SNP analyses. And then one part of this work package is to find out candidate genes for frost tolerance. And we have selected significant SNPs from association mapping studies for winter hardiness, and this was done by RISA's partners from Canada, Monica Boga and Ravindra Chippar, and also we have selected these significant SNPs from other publications. And then we have done these gene annotations from this LO7 genome, which has been sequenced. And one part of this is also that we will develop this kind of cusp markers for the selected SNPs there in Germany. And these cusp markers can be then used in marker-assisted selection in rye breeding. And why we have used or want to use these cusp markers, it's because they have lower costs and they have better genotyping efficiency, and breeding companies, for breeding companies, it's easier to use. And in this method, there are this kind of 2 different forward primers with different alleles and fluorescent dyes, and 1 common reverse primer. And then this fluorescence signal is detected. But at the moment, we don't have these cusp markers, all of them yet available, but I think we will get them in this month. And the other project where we used Rye Landraces was this Microbial biodiversity in Finnish rye sourdough. And this was Luke's project. And our goal was to study genetic diversity of rye landraces and old cultivars, and to better understand rye bread as a functional food. Then we wanted to study diversity of microbiota of these Finnish rye sourdoughs and flour. And also we wanted to study microbial dynamics of those selected sourdough samples when we were doing this, making these breads. And then we wanted to analyze also secondary metabolites from those samples, because we wanted to know which kind of differences is in the beneficial compounds in those samples. And also these food maps were also evaluated.
Let's say your time was almost out.
Okay, this is the last thing, I think. And we used for the analysis of genetic diversity here, these microsatellites, 10 microsatellites were used. And in this study, we had 30 single plants per local rye population. And then we made this kind of dendrogram from these results. And we also checked the heterozygosity level and level of genetic variance within and between the populations. But all of those results have not been published yet, but we are doing manuscripts at the moment. Okay, the last one was also we checked these the diversity of lactic acid bacteria and yeast. So we were sequencing the samples also there in Turku in Finnish Functional Genomics Center. And also some isolates and strains were identified here in Luke. And that was all. Thank you. Do you want